Welcome to BioRatna. Those of you who are watching me for the first time, let me introduce myself. I am Chandana Sengupta, having 18 plus years of teaching experience. I am honored to be recognized as Excellent Educationist by HRD Ministry, Government of India. Few of my suggestions have been incorporated in Standard 11th and 12th NCERT Biology. Many of my students have taken admission in government medical colleges, and every session for 12th board I have given my bio toppers with 98-99 marks. But my real introduction is that I love my subject, of which I am a continuous learner, and I love and care for my students. So students, without much ado, let us start with today's topic. And today's topic for discussion is palindromic sequence. Before seeing what is palindromic sequence, let us first see what is a palindrome. The palindrome. The example given in NCERT is Malayalam. M-A-L-A-Y-A-L-A-M. Malayalam. This word, whether you read it from left to right or you read it from right to left, it spells the same. That is M-A-L-A-Y-A-L-A-M. See from this side, M-A-L-A-Y-A-L-A-M. These kind of words or these kind of sentences are called palindromes. But in molecular biology, it is a bit different. Let us see. Let us take the example of one nitrogen-based sequence in one strand of DNA, which is G-A-A-T-T-C. G-A-A-T-T-C. This is 5' end and this is 3' end. Fine. Okay. Read it from left to right. This is G-A-A-T-T-C. Now read backwards. This is C-T-T-A-A-G. Not a palindrome. Both read different, but you know that DNA is not single-stranded. DNA is double-stranded, and both the strands have opposite polarity. So here I have written 3'. This side I am writing 5'. And the nitrogen bases, they are complementary. So this is C, this will be T, T, A, A and G. Okay. This strand also, you have to keep the polarity same. This side it is 5, this side it is 3. G-A-A-T-T-C. So that's a palindrome. That's a palindromic sequence. So even if you read from 3 to 5, here also you read from 3 to 5, then it is the same. Like C-T-T-A-A-G, this also C-T-T-A-A-G. These are the palindromic sequences. So what you have seen, what is a palindromic sequence? It is the sequence of nitrogen bases. It is the sequence of nitrogen bases on one strand or two strands, on both strands of DNA. Both strands of DNA which read same when the polarity is kept same. Which read same when polarity is kept same. This is the most important point here, that you have to keep the polarity same. Then both read same. This sequence, this palindromic sequence is that of a restriction enzyme from EcoRI, that is EcoRI. Every restriction enzyme has its own palindromic sequence. हर किसी का अलग अलग होता है. There are so many restriction enzymes. Each and every one has its own sequence. Now this sequence, palindromic sequence, is also called recognition sequence. This is also called recognition sequence. Who is recognizing it? Recognition sequence. Who is recognizing it? The restriction enzymes. The restriction enzymes, while cutting the DNA, those restriction enzymes which do not cut at any random place. These restriction enzymes, what they do, they inspect the whole DNA first and they see whether they are finding out that sequence like EcoRI. If this sequence is not present in the DNA, then it is not going to cut. Like that. One peculiar thing about the restriction enzymes is that they do not cut the palindromic sequence from the center. Idhar 3 hai, idhar 3 hai. They will cut on the both strands between the same nitrogen bases. If here it is cutting between G and A, here also it will cut between G and A. This site where it is cutting, this is called restriction site. This site where it is cutting, this is called restriction site. This is also called cloning site in case of the vectors, because this is the place where the vectors are cut and we are putting the desirable gene of which we need the multiple copies. That's why it is called restriction site or the cloning site. Now one more important thing. Let me tell you the palindromic sequence for BamHI. For BamHI and how to remember it easily. Only changes you have to make from the palindromic sequence of EcoRI is that ये जो both sides से जो सेकेंड वाला बेस पेयर है ना, इसे आपको चेंज करना है. चेंज करके क्या करोगे? सेम लाइक द प्रीवियस वन. इस साइड दिस इज G, सो दिस वन, दिस A will become G. This is C, this will become C. This side also do the same thing. This is G and this is C. And here your BamHI palindromic sequence is ready. So what points you have seen? That a palindromic sequence is the sequence of nitrogen base. Nitrogen base का sequence है on both strands of DNA which read same when the polarity is kept same. In the next video, we will find out what is sticky ends and why we need the same restriction enzyme to cut the DNA of the vector DNA as well as the foreign DNA. I hope you have liked this video. Then do subscribe and hit the bell icon so that you get all my notifications. Thank you for watching the video. See you.